A very important scientist was born in Owasso, Michigan. He graduated from Michigan State in 1930, and in 1934, he graduated with his PhD in the Department of Bacteriology. After he graduated, he took a job at the Washington University School of Medicine and started his study on bacteriophage. Bacteriophage is a virus that infects and replicates within bacteria and archaea. He started and established the American Phage Group, which had a tremendous influence on bacteriophage research. This is a picture of him when he first started out at the Washington University School of Medicine. When asked what his idea of happiness would be, he replied, "To have an experiment that works and do it over and over again." This showed that he really wanted his experiment to work and to show the world. And his main goal was to demonstrate that DNA, rather than proteins, is the carrier of genetic information. His work appeared to be well supported and was endorsed by other researchers and scientists. The Avery Group was first molecular biologists and pioneers in immunochemistry. This is a picture of the basic blending of what he was trying to find out in bacteriophages. He needed to be able to examine different parts of the phages they were studying. He needed to do this separately, so they needed to distinguish the phage subsections. Viruses were known to be composed of a protein shell and DNA, so they chose uniquely label each with a different elemental isotope to be able to tell them apart and look at them separately. This allowed each to be separately observed and analyzed by the scientists. This is a picture of a common bacteriophage. He inserted the radioactive elements in the bacteriophages by adding the isotopes to separate media within which bacteria were allowed to grow for 4 hours before bacterial introduction. His experiment concluded that DNA, not protein, was a genetic material. Protective protein coat was formed around the bacteriophage, but the internal DNA is what conferred its ability to produce progeny inside a bacterium. This is a picture of basic DNA genetic material which he was trying to find out that carried it. There was no specific conclusions can be made regarding whether material that is sulfur-free enters a bacterial cell after phage absorption. Further research was necessary to conclude that it was solely bacteriophage's DNA that entered the cell and not a combination of protein and DNA where the protein did not contain any sulfur. This is a picture of the Hershey-Chase experiment, which is his most famous. In 1969, he won the Nobel Peace Prize, which he shared with his partner for psychology and medicine. In 1962, he became the director of the Genetics Research Unit at Cold Springs Harbor Laboratory. In 1974, Hershey retired, though he was still a regular visitor at Cold Springs Harbor Laboratory. A building was built on his behalf and dedicated to him, which finished off his scientific career. Thank you.